Let me give a quick introduction about the college. KGRL College of Pharmacy is established in 2007 under the aegis of the BHIMAVARAM Education Society with the main objective of providing world-class education in pharmacy with a mission to make it a career of excellence, ensuring high quality and value-based education and unwavering commitment to provide quality teaching and innovative research by transforming young talented students into global pharmacists. KGRL College of Pharmacy is an elite institution with the state-of-the-art infrastructural facilities, modern laboratories, and student support services coupled with dedicated and experienced faculty members. And the principal, the holistic academic culture, individual care to the students, good academic ambience, campus facilities, commitment for quality education has attracted the attention of aspiring students to nurture and shape their career in field of pharmacy. To welcome is to show honor. To welcome is to establish integrity. As this first national conference is built with the intent of active and interactive learning organized by KGRL College of Pharmacy. The one who acts as a pillar of support and strength, his idea of student centricity have assured in several developments towards strengthening student employability, support spirit, achievements among many other noteworthy to be mentioned here. I heartily welcome Dr. K. Nageshwar Rao Garu, Director, KGRL College of Pharmacy. May I request Vani Pravalika to welcome sir with a bouquet. Now the time has arrived to add some more dignity and fragrance to the dais. I extend a warm welcome to Dr. K. Venkatramana Garu, President APTI, AP State Branch, Principal ASN College of Pharmacy, Tenali. And I request Shravani Durga to present a flower bouquet. We welcome you, sir. I am blessed to welcome Dr. A. Lakshman Rao Garu, Vice President, APTI, AP State Branch, Principal, VV Institute of Pharmaceutical Sciences, Gudlavalleru. May I request Deepika to welcome sir. Next, I take the privilege to welcome Dr. D. Narendra Garu, Secretary, APTI, AP State Branch, Principal, VJS College of Pharmacy, Rajahmundry. It's my request for mercy to present a flower bouquet. I take pleasure to invite Shri Kotikalpudi Govindarao Garu, President, KGRL College. Next, I take privilege to specially welcome Shri Mente Ram Manohar Garu, Secretary and Correspondent, KGRL College. May I request Vani to welcome sir. Next, I take this opportunity to invite Dr. Sridhar Babu Garu, Principal, Shivani College of Pharmacy, Warangal. May I request Shahin Jaha to offer a bouquet? Next, I take this opportunity to invite Dr. K. Natras Garu, Principal, Sri Vishnu College of Pharmaceutical Sciences. I request Sirisha to offer a bouquet. Next, I take this opportunity to invite Dr. K. Natras Garu, Principal, Sri Vishnu College of Pharmaceutical Sciences. I take this opportunity to invite Dr. K. Prasad Garu, Senior Professor, Sri Vishnu College of Pharmaceutical Sciences. I request Rajeshri to present a bouquet. I take this opportunity to invite Dr. K. Prasad Garu, Senior Professor, Sri Vishnu College of Pharmaceutical Sciences. I request Rajeshri to present a bouquet. Next, I take the privilege to welcome Dr. Vinod Rao Garu from the Department of MBA, KGRL College. I request Mercy to present a flower bouquet. I am profoundly delighted to take an opportunity to introduce one of the resource persons for the day. He is none other than Dr. Sandeep Kumar, Assistant Professor, Department of Regulatory Affairs, Naipur, Hyderabad. Again, I request Madhavi to present a flower bouquet. It's my pleasure to invite the other resource person for the day, Shri Fatru Patel, Regulatory FI Specialist, Dr. Reddy's Laboratories Limited, Hyderabad. I request Sirisha to offer a bouquet. I take this opportunity to invite Dr. D. Raghava Garu, Principal, KGRL College of Pharmacy. I request Shahin Jaha to offer a bouquet to sir. Light symbolizes victory over darkness and defeat ignorance. It marks the beginning of auspicious moments and events. I request the dignitaries to do the honor and I request the audience to rise for the same. Now I would like to invite Shri Mente Ram Manohar Garu, Secretary KGRL College, to speak a few words about the conference.
Respected Director Garu, Nageswara Garu, K. Nageswara Garu, respected Principal, respected Director K. Nageswara Garu, KGRL Pharmacy, respected Dr. Raghava, Principal KGRL Pharmacy, and for all the dignitaries who have come here to share their knowledge regarding the seminar of the morning, Global Pharmaceuticals Regulatory Affairs. I invite Dr. Sandeep Kumar, Assistant Professor, Nipur, Hyderabad, Sri Patru Patel, Dr. Reddy Lab, K. Venkataraman Garu, President APTI, Dr. A. Lakshman Rao Garu, Vice President APTI, Dr. D. Narendra, Secretary APTI, Dr. Sridhar Babu Garu, Principal Shivani College of Pharmacy, and also invite Dr. K. Nataraj, Principal SVCP, and also Dr. K. Prasad, Senior Professor SVCP. I cordially invite each and every one of the students and also the dignitaries on the dais on behalf of KGRL College and Bevar Education Society. Welcome, sir. Today we are eagerly waiting for your words on global pharmaceuticals regulatory affairs. So many students from our college and from the adjoining places of BHIMAVARAM have come here. I cordially welcome each and every student on behalf of KGRL Pharmacy. Let's today listen the words of the dignitaries and share their knowledge for upgradation in your careers and to have a better life and settle in a better careers. Pharma has been in demand now. You have chosen a good career. Pharma was in demand now and its opportunities were in abundance, even in India and abroad. During COVID-19, we have seen the working of pharma people and their efficiencies and work, and they were in great demand in providing the patents and in manufacturing the vaccines. Let's today concentrate on topic global pharmaceuticals regulatory affairs. What is regulatory affairs? I have been discussing with the specialists here and have given certain inputs. It's a unique mix of science and management to achieve a commercially important goal within a drug development organization. Touches everything related to drugs from the non-earliest clinical studies to development into routine manufacturing and marketing, can add significant impact for patients and drug companies. Why is regulatory affairs needed? Drug development and commercialization is highly regulated. The path of drug registration is paved to good intention but can be complicated. Things change constantly. And what are the parameters for regulatory affairs? It's the design, coordination, construction, testing, and drug regulation, natural laws, regional laws, national and regional guidelines, and also international guidelines. These all things in elaborate will be given by the specialist people who have come here, all from Hyderabad and adjoining places. And once again, welcome each and everyone. And they are going to give you an elaborate discussion on the topics. You can share their knowledge and enlighten yourselves and clear your doubts so that you can lead a successful path in your lives. Thank you. Thank you very much.
Thank you, sir, for your valuable words. Now I request Dr. K. Nageshwarao Garu to speak a few words about the conference.
Good morning, everyone. So, respected dignitaries, resource persons, other distinguished faculty members, principals of various colleges, and all the delegates, those who have come over here for attending the seminar, and we have arranged a seminar on global drug regulatory affairs, and we call one academician and one industry person, one NIPER and Dr. Reddy's Laboratories. They are going to give the elaborate subject on the particular topics. So the purpose of meeting to enlighten the present scenario, what is happening and what is required for the industry. So based on that, we have chosen this subject in association with APTI, Association of Pharmaceutical Teachers of India, AP State Branch. Now each and every faculty member must be a APTI member. Okay, so all colleges are being enrolled in APTI as institutional members of the APTI, so that there are a lot of benefits, and APTI as a faculty member, it will take care of all the faculty members. APTI will take care, and in association with APTI, we are arranging the program and to inculcate a good knowledge. So every month or every quarter, being arranged in different parts of the colleges in throughout the state. So try to enlighten yourself by joining or participating. Don't expect, see 200 rupees or 300 rupees, nothing for you. You may be spending 400 or 500 rupees for one show. So try to, I advise all the people, faculty members as well the students, to spare some time and enroll yourselves in the meetings or seminars being arranged by different colleges in different places. Try to enlighten, register yourself and enlighten yourself, and try to see now PCI, Pharmacy Council of India, is going to do a lot of changes in the curriculum also. So whatever the certificates you are having, there may be giving some credit points for this attending seminars also. Suppose today you have taken the certificate from this college. So accumulate all the things and you will be awarded some credit points for that also. It is being implemented by PCI. So try to, don't think that what is the advantage of attending the seminar. So having attended the seminar, and you will be having certificate, and that gives you some credit points for your academic calculations. This SGPA, CGPA calculations, it will be helpful for you. So I sincerely advise all the people to spend your time, comfortable stay, and learn the things what the resource persons are giving or imparting to you. Try to have interaction with the session, have interaction, try to learn and try to get your answers. So I advise all people to spend this time, and first of all I congratulate all people for coming over here from the different places on the request of our people. And Dr. Nagarjun Reddy has taken the initiative, and Dr. Ramarao, our faculty members, they have visited each and every college of Krishna district, Guntur, East Godavari, West Godavari, invited. Okay, so thanks for attending this program, and happy to announce that about 300 students from other colleges, East Godavari, West Godavari and Krishna, and about 300 students of ours. So totally it comes to around 700 members. So there may be some laggings. So try to adjust yourselves, because we don't know till last night what is the strength of the people coming from outside. Okay, anyway, we are extending our hospitality. See, whatever you do, hospitality must be good. Hospitality. Okay, so try to adjust yourselves. If any some lacuna is there, some deficiencies are there, try to adjust. This is treat that this is college or this campus is yours. So we are going to help you in all aspects, hospitality arrangements, etc. Try to spend your time for today. And what are the posters are this one, e-posters are the presentations are there. Try to, after the plenary sessions, that will be taken care. So afternoon session, after lunch, it will be arranged, presentations, etc. And around 3 o'clock, we'll start the valedictory function. Till that time, each and every student will be eagerly waiting. What is my position, first prize or second prize or whatever it is. So try to carry. So prizes, 700 members are there, 700 members will not get. But 3 or 4 items are there, each and every item about first prize, second prize, third prize, like that. So have certificate, participation certificate and etc. And try to have comfortable stay and try to learn and leave the campus with fruitful results. Wish you all the best. Wish this program a grand success. I congratulate all the stakeholders like Dr. Nagarjuna Reddy, who has taken initiative, Dr. Ramarao, Dr. Edward Razu, and other people, those who have taken active participation. See, I wanted to say one thing. I am just only a spectator only. I am playing as a spectator. But only thing is, everything is being organized by those people. So that leadership qualities one should develop. And Dr. Lakshman Rao is there, and Dr. Venkatramana is there. They are all very close to me. And they are invited, asked for that. So I voluntarily accepted this date. Though I have got some other programs, I cancelled other programs and made this program arranged today. So I wish this program a grand success. I congratulate for accepting this our invitation, resource persons and other dignitaries. Thank you very much, and wish this program a grand success. Thank you.
Thank you, sir, for your helpful comments. Now I would like to invite Dr. K. Venkatramana Garu for his greetings.
Good morning, respected Chairman of KGRL Group of Institutions, Sri M. Ram Mohan Rao Garu, and Director of KGRL Pharmacy College, Dr. K. Nageswara Garu, Principal of the institution, Dr. D. Rao Garu, and Convener and Coordinator of this program, Nagarjuna Reddy Garu, and my dear APT Office Bearers Vice President Dr. A. Lakshman Rao Garu, APT Secretary Dr. D. Narendra Garu, and resource persons Sandeep Kumar Garu and Patru Patel Garu, and my office bearers EC member Dr. K. Nataraj Garu, former Principal of Vishnu College of Pharmacy Dr. Prasad Rao Garu, Sridhar Garu, came from Telangana state, Warangal, and all the dignitaries on the dais and off the dais, and various college principals and faculty members and students in this institution and outside the institutions came from far away, from BHIMAVARAM. First of all, I congratulate all the students for your interest and attending this today seminar, Global Pharmaceutical Regulatory Affairs, in association with APTA AP State Branch. First of all, I thanks to management and director and principal of the institution giving this opportunity. So APTA associate with your institution, particularly in the nowadays the students, the global pharmaceutical regulatory affairs, what is going on in India, across the India. So you people all know very well, pharmacy is a evergreen profession. ఈ మనిషి భూమి మీద బతికి ఉన్నంత కాలము ఫార్మసీ అలాగే ఉంటుంది సజీవంగా. ఎనీ అదర్ category of the jobs or whatever it may be, some fluctuations is arises already started in the US, Canada, European countries in the software already started. Our profession is evergreen profession. That is the witness to identified persons, to identified companies prepared the vaccines. Serum Institute one, Bharat Biotech. First of all, we are proudly Telugu వాళ్ళు ఇండియన్ మనవాళ్ళైనటువంటి డాక్టర్ కృష్ణ ఎల్లా గారు భారత్ బయోటెక్ కోవాక్సిన్ అనే వ్యాక్సిన్ తీసుకుని వచ్చి భారతదేశంలో ఎంతోమందికి ఇవ్వటం జరిగింది అది ఈ కోవిడ్ కండిషన్స్లో. అలాగే సెరమ్ ఇన్స్టిట్యూట్ నుంచి కూడా వ్యాక్సినేషన్ రావటం జరిగింది. సో అవర్ beloved Prime Minister of India supported 138 crores peoples. ఈ వ్యాక్సినేషన్ చేయటం వల్ల ఈ రోజు నాడు భారతదేశం సజీవంగా ఉండగలిగింది. మనకంటే అడ్వాన్స్డ్ ఉన్న టెక్నాలజీ ఉన్న దేశాలు ఆల్రెడీ అగైన్ స్టార్టెడ్. యుఎస్లో కానీ చైనాలో కానీ కొన్ని దేశాలు ఆల్రెడీ, కొరియాలో కానీ కొన్ని దేశాలు ఆల్రెడీ అగైన్ కోవిడ్ వచ్చేసింది. మనం ఎందుకని ఇలా ఉన్నామంటే మనం తీసుకునే ఆహారపు అలవాట్లు, మనం ఉన్న కండిషను, మన భారతదేశ సంస్కృతి, కట్టుబాట్లు, అలవాట్లు అన్నీ, కంప్లీట్ ఫుడ్ అన్నీ కూడా ఎవ్రీథింగ్ పాజిటివ్ ఉండటం వల్లే మనం హ్యాపీగా ఈరోజు ఉండగలిగాము. సో దీని వెనక ఒక వ్యక్తి కాదు, ఒక వ్యవస్థ. యూ పీపుల్ నో వెరీ వెల్ ఇన్ కోవిడ్ 2 ఫస్ట్ అటెంప్ట్ అండ్ సెకండ్ అటెంప్ట్ రెండిట్లో కూడా మనం ఎన్నో లెసన్స్ తీసుకున్నాం. డాక్టర్స్తో పాటు ఆన్ పార్ విత్ ఫార్మాడీ స్టూడెంట్స్ అలాంగ్ విత్ ఫార్మసీ, బీఫార్మసీ ఆర్ డిప్లొమా ఇన్ ఫార్మసీ, మాస్టర్ ఆఫ్ ఫార్మసీ, ఈ అందరూ కూడా సమైక్యంగా యాజ్ ఏ ఫార్మసిస్ట్గా వాళ్ళ యొక్క డ్యూటీస్ వాళ్ళు చేయటం వలన 24/7 చేయటం వల్ల ఈరోజు నాడు ఫార్మసీ రంగానికి, భారతదేశంలో ఫార్మసీకి ఇదివరకు ఉన్నది 70 సంవత్సరాలు బట్టి, 72 సంవత్సరాలు బట్టి, కానీ కోవిడ్ 2 ఇయర్స్లోనే ఫార్మసీ అనేది తీసుకెళ్ళి ఒక స్టేజ్లో తీసుకెళ్ళిపోయింది ఇండియాని. కారణం ఏంటంటే ఈ వ్యాక్సిన్స్ తయారు చేసిన దాంట్లో కానీ, డాక్టర్స్తో పాటు సపోర్ట్గా ఉన్న ఫార్మసిస్టులు కానీ, దానివల్లనే మేజర్గా మన ప్రొఫెషన్కి ఎక్కువగా ఇంపార్టెన్స్ అనేది బాగా ప్రతి వాళ్ళకి పల్లె దాకా వెళ్ళిపోయింది. ఇదివరకు లేదు. ఫార్మసీ అంటే ఏదో మందులు ఇస్తారంట, మందులు అమ్ముతారంట అనుకున్నారు. కానీ అలా కాదు. ఇది వీళ్ళు చేసే డ్యూటీ, ఇది వీళ్ళు చేసింది అనేది మనకి స్పష్టంగా తెలిసింది. ఈ కోవిడ్ సిచ్యువేషన్లోనే బాగా బయటికి ఎలివేట్ అయింది. గవర్నమెంట్ ఆఫ్ ఇండియా కూడా డిఫరెంట్ టైప్ ఆఫ్ ద రెగ్యులేషన్స్, రెగ్యులేటరీ బాడీస్, వాట్ ఈజ్ గోయింగ్ ఆన్, వాట్ టైప్ ఆఫ్ ద యాక్ట్స్ వీఆర్ గోయింగ్ టు ఇంప్లిమెంట్, ఏమి చేయాలి, ఎలా చేయాలి అనే దాని మీద ఎప్పటికప్పుడే మొత్తం కూడా కొత్త అన్నీ బయటికి తీసుకురావటం వలన ఇన్ దట్ పర్టిక్యులర్ వ్యూ మీద టుడే గ్లోబల్ ఫార్మస్యూటికల్ రెగ్యులేటరీ అఫైర్స్ న్యూ what is going on as a present status in India, what is the regulatory bodies, acts, how it helps to the pharmacist and as well as the companies as well as the patient oriented. So ఆ వ్యూలో ఏ విధంగా పనిచేస్తున్నాయి, ఏ విధంగా ఉంది అనే దాని మీద ఈరోజు 2 eminent speakers invited, one from Naipar, Hyderabad, one person from Dr. Reddy Laboratories. So బికాజ్ మీరు యాజ్ ఎ స్టూడెంట్కి వెళ్ళి వారితో మీట్ అవ్వలేరు. కానీ ఇలాంటి ప్లాట్ఫామ్లో డెఫినెట్గా ఇది ఒక మంచి ఆపర్చునిటీ. ఈ ప్లాట్ఫామ్లో మీకున్న సందేహాలు వారు చెప్పిన తర్వాత తెలుసుకోవటము, ఏ విధంగా ముందుకు వెళ్ళాలి అనేది డెఫినెట్గా మీకు ఇక్కడ యూస్ఫుల్ అవుతుంది. సో ఆ వ్యూలో మీరు అందరూ కూడా స్టూడెంట్స్ తప్పనిసరిగా ఈ 2 లెక్చర్స్ చక్కగా విన్న తర్వాత నెక్స్ట్ యాజ్ యూజువల్ ప్రోగ్రాంలో ఈ పోస్టర్ ప్రెజెంటేషన్లో మీరు పాల్గొంటారు. సో మై డియర్ యంగ్ బడ్డింగ్ ఫార్మసిస్ట్, ప్రపంచం అనేది ఎక్కడో లేదు, మీ చేతుల్లోనే ఉంది. ఆపర్చునిటీ అనేది ఎక్కడో లేదు, అది మీ ద్వారానే ఉంటుంది, మీ మీదే ఆధారపడి ఉంటుంది. అది ఆ విధంగా ఇక్కడ ఉన్న అవకాశాలని అందిపుచ్చుకొని తప్పనిసరిగా ఐఆమ్ ఇండియన్ అని ప్రౌడ్గా చెప్పుకోగలిగే విధంగా మీరు ఎస్టాబ్లిష్ అవ్వాలి. దానికి తగ్గట్టుగా కాలేజీలు, దానికి తగ్గట్టుగా ఫెసిలిటీసు అన్నీ ఉంటాయి. పేరెంట్స్ సహాయము, కాలేజీల సహాయము తప్పనిసరిగా ఉంటాయి. దాన్ని అందిపుచ్చుకుని మీరు చక్కగా మంచి ఉన్నతమైన స్థితిలో ఉండాలని ఏ కళాశాల అయినా సార్ గారు కానీ, చైర్మన్ గారు కానీ, డైరెక్టర్ గారు కానీ, ప్రిన్సిపల్ గారు ఏ కాలేజీలైనా తప్పనిసరిగా కోరుకునేది ఇదే. మీరు కాలేజీకి ఏమి ఇచ్చారని కాదు, మీరు ఏమి చేశారు, దేశానికి మీరు ఏమి చేశారు, మీ వలన ఏంటి. సో దానికి మీరు గట్టిగా ప్రయత్నం చేయాలి, ట్రై చేయాలి. ఇలాంటి వేదికని తప్పనిసరిగా ఉపయోగించుకొని ఇంకా మరిన్ని ప్రోగ్రామ్స్ చక్కగా పాల్గొని మంచి నాలెడ్జబుల్గా మీరు ఉండాలని చెప్పి, అలాగే ఈ యొక్క అవకాశాన్ని ఇచ్చిన మేనేజ్మెంట్ వారికి, డైరెక్టర్ గారు, ప్రిన్సిపల్ గారు, కన్వీనర్ ఆఫ్ దిస్ ప్రోగ్రామ్, అలాగే ప్రత్యేకంగా టీచింగ్ అండ్ నాన్ టీచింగ్ ఆఫ్ దిస్ ఇన్స్టిట్యూషన్. ఈ ప్రోగ్రామ్ స్టార్ట్ చేసిన తర్వాత నేను ఆల్రెడీ డాక్టర్ నాగేశ్వరరావు సార్ గారితో మాట్లాడుతూ ఉన్నాను. అందరూ కూడా చాలా రోజులు బట్టి ఈ ప్రోగ్రామ్ మీద ఎంతో కష్టపడి పనిచేశారు. అందరూ కూడా అందరికీ కూడా పేరు పేరున ధన్యవాదాలు తెలియజేస్తున్నాను. ఎందుకంటే ఇలాంటి ప్రోగ్రామ్ అరేంజ్ చేయటువంటి నాటే ఇన్ ఈజీ టాస్క్, చాలా కష్టమైన పని. సో ఎంతో ప్రణాళికాబద్ధంగా ప్రిపేర్ చేయటము, అందరినీ జాగ్రత్తగా రిసీవ్ చేసుకుని మళ్ళీ అందరినీ జాగ్రత్తగా పంపించడం అనేది నాట్ ఈజీ. మీరు అనుకుంటారు పిల్లలు రూపాయలు కట్టిస్తాం మన ఇల్లు వచ్చేసాం అంటారు, కానీ అలా ఉండదు. చాలా చక్కగా మీకు మంచి స్పీకర్స్ని తీసుకురావాలి, మంచిగా చెప్పించాలి. మీరు దాని నుంచి విషయం తెలుసుకుని మీరు హ్యాపీగా ఇంటికి వెళ్ళాలి. సో ఆ వ్యూలోనే ఎక్కడైనా సరే ప్రోగ్రామ్ అనేది చక్కగా చేయటం, చక్కగా అన్నీ ప్రతి ఈవెంట్లో కూడా పాల్గొని చక్కగా హ్యాపీగా వెళ్ళాలని చెప్పి నేను మనస్ఫూర్తిగా కోరుకుంటున్నాను. థ్యాంక్ యూ.
Thank you, sir, for enlightening us with your words. Now I request Dr. A. Lakshman Rao Garu to speak a few words.
Respected management of KGRL College of Pharmacy, Sri M. Ram Manohar Garu, and my well-wisher and good, just like a friend, Director of KGRL College of Pharmacy, Dr. K. Nageshwara Garu, and my beloved student and Principal of KGRL College of Pharmacy, Dr. Raghava, and my crew of APTI AP State Branch, respected President of APTI AP State Branch, Dr. K. Venkatramana Garu, and respected Secretary of APTI AP State Branch, Dr. D. Narendra Garu, and the resource persons for today's event, Sri Fadhu Patel Garu, Dr. Reddy's Laboratories Limited, Hyderabad, Dr. Sandeep Kumar Garu, National Institute of Pharmaceutical Education and Research, Hyderabad, and other guests, invitees, principals, teachers, and student fraternity. A very good morning to all. On behalf of Association of Pharmaceutical Teachers of India, APTI AP State Branch, a warm welcome to all of you for the first national conference on global pharmaceutical regulatory affairs. Nowadays, the regulatory affairs is playing a very vital role in the healthcare industry, because the regulatory affairs, especially if you can take all the multinational companies, they are having a own department of regulatory affairs division. And because of this particular point of view, and I especially congratulate the Department of Regulatory Affairs, KGRL College of Pharmacy, for their keen interest for organizing this particular wonderful event with the theme of Global Pharmaceutical Regulatory Affairs. And dear students, and these type of seminars, conferences and workshops are really so much helpful for the student community and to gain a lot of knowledge and to enrich yourself for upgrading your knowledge. And with this particular aspect, and APTI, Association of Pharmaceutical Teachers of India, is planning to conduct various events in the state of Andhra Pradesh. Our team of APTI AP State Branch is putting lot of efforts for organizing the various seminars, conferences, workshops, training programs and faculty development programs and all the things for the benefit of the teachers as well as the student community. And nowadays, we know very well that the pharmaceutical regulatory affairs is having a wide importance in the entire world. Okay, and if you can have a knowledge on the regulatory affairs, you may get a lot of opportunities are there, not only in India, in abroad also. Regulatory head, like that, every kind of multinational companies having their own divisions in the various countries. With this particular theme, I requested the two people. One is Padru Patel and second one is Dr. Sandeep Kumar. Really, on behalf of this particular platform, I really thank you very much, sir, for accepting the my request for coming to this particular conference and for sharing your knowledge for the benefit of student fraternity members. And next, another important aspect in this case is, especially my dear student friends, now the world is in your hands and it's your time to upgrade yourself and to gain the update knowledge. And what are the various prominent topics in the area of pharmaceutical sciences? For example, if you can take the global regulatory affairs and also 3D printing and also nanotechnology and also artificial intelligence and also chronotherapeutics, like that, so many novel topics are there in the area of pharmaceutical sciences. As a pharmacy student, as a budding pharmacist, you must have a sound knowledge in all these particular topics. Definitely, you may have a very bright future in your particular future endeavors. And on this particular platform, I especially thank the principal, Dr. D. Raghava, and the head of department of this particular Department of Regulatory Affairs, Nagarjun Reddy and Ramarao and Edward and other crew of the KGRL College of Pharmacy for their meticulous planning and also with the support of all the teaching and non-teaching members of KGRL College of Pharmacy. Okay, with all this particular teamwork only the event is going to be grand success. On this particular occasion, I specially congratulate Dr. K. Nageshwara Garu. Under his dynamic leadership, the KGRL College of Pharmacy is growing day by day enormously, and I especially thank you for your interest, sir, for developing the organization, because he is putting lot of efforts in getting the new courses and establishment of the new courses. And recently he initiated the diploma in pharmacy also, and they make it as a successfully they got successful admissions in this particular college and Department of Regulatory Affairs, like that. He is showing his mark in development of KGRL College of Pharmacy. Okay, and I especially thank the KGRL College of Pharmacy entire team for giving an opportunity to organize this particular first national conference in association with APTI AP State Branch. Thank you very much, sir, for giving an opportunity to work together for making this particular event for the benefit of the student fraternity. And I finally wish this particular first national conference on global pharmaceutical affairs organized by KGRL College of Pharmacy in association with APTI AP State Branch will become a very grand success. Thank you one and all for giving this wonderful opportunity.
Thank you, sir. Thank you for your encouraging words. Now I request Dr. D. Narendra Garu to speak a few words about the conference.
Good morning, one and all. Respected dignitaries on the dais, dear faculty members of KGRL College of Pharmacy and other colleges who came to attend this national seminar. I would like to thank the management of KGRL College of Pharmacy for conducting this seminar in association with Andhra Pradesh State Branch APTA. So I am also thankful to the students who have registered and who are contributing something for the welfare of teachers of pharmacy colleges in Andhra Pradesh state. The registration fees is very less when compared to other seminars and conferences that you are going to attend. As our director Nageswara Garu said, it is like one movie ticket. So out of this, the institution is going to contribute something to Association of Pharmaceutical Teachers of India, Andhra Pradesh. That money, what you are getting from the conferences with support of the institutions who are going to organize these events, is particularly spent only for the betterment and welfare of teachers who are in need of money. When the time comes, the association will be with them and we will support them by organizing these events. So hope the students' contribution will be more in future conferences, and request the students to be associated with the conferences and seminars that are going to be conducted in our Andhra Pradesh state, in each and every corner of the state by Association of Pharmaceutical Teachers of India, Andhra Pradesh State Branch, under the leadership of Dr. K. Venkatramana Garu, who is the President of the state APTA, and Vice Presidents Dr. Y. Ankam Chaudhary Garu and Dr. A. Lakshman Rao Garu. Myself, I am the Secretary, and Office Bearer Nataraj sir is also here, and we have some more Office Bearers team. We are here present. Maybe you are a student, in future you might be our colleague. So it is a teacher-student interaction, so we should get the support from the students, as we are giving our wholehearted support to the student for his achievements and reaching his dreams. So I request all the students to be associated with APTA for the betterment of our profession. If the teacher survives, the profession survives. So hopefully we have attended a wonderful national seminar. Nageswara sir is not only the well-wisher of Lakshman Rao sir and others, he is the well-wisher of pharmacy profession. So I congratulate Nageswara Garu for this good gathering, and I congratulate Raghav Garu. Hope under his principalship this is the first national seminar that he is going to organize today. And I congratulate Nagarjuna Reddy Garu and Ramarao Garu and also other team of KGRL College of Pharmacy. So I request the students to utilize this opportunity. So money is yours, time is yours. If you learn something, it will be useful for you. If you have some snaps of subject, it will be utilized for you. If you have snaps of your own face, it's of no use. So I request, instead of selfies, you please get some knowledge in this campus, and hopefully the resource persons from Reddy Laboratories and NIPER have enough talent to explain each and every aspect of the regulatory affairs. And also the presentations by the students will also enlighten you the thoughts in various aspects of pharmaceutical sciences. And I once again congratulate the faculty members behind the conduct of this conference. We know the pain and pressure of the faculty members in organizing such type of activities. So we should congratulate the faculty members for this success. And also Nageswara sir is known for his hospitality. He always bothers about hospitalities, and we had good breakfast, and we all hopefully we are going to have wonderful lunch here. Okay, he will arrange snacks, tea, everything. If you won't eat also, he won't leave you. So it is your responsibility to add his hospitality in this campus. And I once again thankful to Raghav Garu for arranging all these things for us. And I thank students also, and want students to participate more seminars and conferences to be organized by Association of Andhra Pradesh Teachers of India, Association of Pharmaceutical Teachers of India, Andhra Pradesh. So thank you, sir. Thank you for giving me this opportunity.
Thank you, sir. Thank you for your valuable words. Now I request Dr. Sridhar Babu Garu to speak a few words.
Dignitaries on the dais, and all the delegates, good morning to all of you. So firstly, I profusely thankful to Director of KGRL Pharmacy College, Dr. Nagesh Rao sir, for inviting me and also making part of this conference. And I am also thankful to Dr. Raghava, who is my student, for inviting me for this conference. And I take this opportunity from this dais to congratulate Nagesh Rao sir and also Raghava and the management of this KGRL institutions to getting new course, diploma course in this year. So this is the great thing. So I always listen to Raghava that they are trying for the getting a new course. So I know the struggle to get the new course by these two people. And secondly, I want to congratulate APTI of Andhra Pradesh. So we don't know earlier, nobody knows about the APTI and also activities of APTI. I was member in 2004 from Warangal, and there is no activities, that means proper activities by the APTI of combined Andhra Pradesh or separated Telangana state and also AP. But recently the APTI elections made something new, or it is somewhat making just like general elections, like MLA elections and MP elections. Myself and also my brother Dr. Raghunandan, we traveled all 4 states, south states, to campaign for the Dr. Sudhir Kumar, chairman of KGRL College of Pharmacy Warangal, and also for Ramada sir, South Vice President, PCI EC member. So we made some vibrations in the elections of APTI, and here I would like to congratulate APTI Andhra Pradesh, because these people, they are making more conferences, more seminars, and also more activities. And morning itself I heard from the D. Raghava that APTI Andhra Pradesh branch is making some insurance. So insurance towards the faculty members, that is the great thing I heard from Raghava. So because of their activities and also their contribution towards the students and also Andhra Pradesh pharmacy, so I want to congratulate all the APTI members like President sir, Venkatramana sir, and Lakshman Rao sir, Vice President sir, and Secretary Narendra sir. So these people are running towards the improvement of the pharmacy profession, as well as to inculcate knowledge towards the students. And so this is about the APTI of Andhra Pradesh. So here today is the regulatory affairs program. So I don't want to explain about regulatory affairs, because you have 2 eminent speakers here. Lastly, I want to say only one thing in respect of the regulatory affairs. One of my students, Premalata, now she is regulatory global head for the company of Takeda. For the company of Takeda in USA, United States of America, and she is junior of your principal, Dr. Raghava. So you see the importance of the regulatory affairs. And one of the other students of Raghava's classmate, so when I was doing my PhD, they were my students, and he said that I am working in regulatory affairs in 2007, 2008, like that. So I asked him that what is regulatory affairs? It is nothing but, sir, it is simple white collar job. he ended with only one sentence, a simple white collar job. So you can see the importance of regulatory affairs division, and the students, you can make yourself to come forward to such type of areas where you can excel your career. So once again, so as Nageshwar sir said that I am representing Telangana state. So I am from Warangal. I am representing Telangana state, and I am humbly thankful to Nageshwar sir for inviting me here and giving this opportunity. Thank you very much.
Thank you so much, sir, for your greetings.
డ్రగ్ రెగ్యులేటర్ అయిపోయి చూడండి, ఇక్కడ మాకు ఉంది బ్రాంచ్ 17 సీట్స్ అయిపోయినాయి. అట్లీస్ట్ 100 మెంబర్స్ ఆర్ దెన్ ద క్యూ. ఎవ్రీబడి ఈజ్ ఆస్కింగ్ నాకు డ్రగ్ రెగ్యులేటర్ సీట్ కావాలి, సీట్ అమ్మ మీకు ఖాళీ లేదు, 10 లక్షలు సీట్ ఇస్తా, 10 లక్షలు పే చేస్తాను, ఐఎమ్ నాట్ ఇన్ ఎ పొజిషన్ టు ఆఫర్ ద సీట్. సో దట్ ద సిట్యుయేషన్ వెన్ ఐ స్టార్ట్ ఇన్ 2014, ఫస్ట్ 3 years no admissions. So drug regulator has got its importance, and everybody is asking for drug regulator affairs seat. టెక్నాలజీ సీట్లు ఉన్నాయి, ఎన్ఆర్సీ ఉన్నాయి సార్, నాకు డ్రగ్ రెగ్యులేటరీ అఫైర్సే కావాలంటారు. ఎంతమందికి ఓన్లీ 17 seats ఆర్ దేర్, 15+2. సో అన్ని 17 సీట్స్ కూడా ఫస్ట్ ఇన్స్టెన్స్లోనే అయిపోయినాయి. మా స్టూడెంట్స్కే ఐ కుడ్ నాట్ ప్రొవైడ్. ఓకే, టెక్నాలజీ సీట్స్ కొద్దిగా 2-3 ఉన్నాయి. బట్ సో డిమాండ్, ట్రై టు అండర్స్టాండ్ ద డిమాండ్, అండ్ ఆన్ ద బేసిస్ ఆఫ్ దట్ వన్ we are choosing the subject, and accordingly we call the experts from the one from academician and one from the industry, so that academia industry, so that both should travel together. Okay, so that is the importance. Try to gain some experience from today's topic and this platform, and learn something and leave the campus with fruitful results. Wish you all the best. And we will have, because plan for first planning session in morning session 11 o'clock. So let us go for the sorry for not giving the opportunity other things. This is your program. So let us give the top priority for the plenary sessions number 1 and number 2. So let us go as per the, sorry for not giving this. Okay, let us give importance for the plenary sessions. Try to enjoy the sessions, please. Thank you. You go ahead, ma'am. So we'll leave the dais.
So now it's time to start the plenary session. So before starting up our plenary session, we have a wonderful classical dance performance by our second year pharmacy student, Bharati. So she's an international classical dancer. So let me invite on the stage M. Bharati of second year B.Pharm to perform a welcome dance.
కలిగేలా ఆత్మకు ఘర్షణం జరిగింది సాగే ప్రతి నిమిషం మసి మన్నున దాగిన కుందనం మరిగాగే జ్వాల शाश्वतम् आदि शाश्वतम्
That was incredible. Thank you, Bharati, for such a beautiful dance performance. A huge round of applause for her.
She is our second year B Pharmacy student, international classical dancer. The moment I saw her, when she has given her performance in the precious day, by viewing that, immediately I was surprised, happy, and immediately I declared her ambassador of this KGRL College of Pharmacy. Now she is the ambassador for KGRL College of Pharmacy. I declared her on the spot, and I offered her, for all your travel abroad or in national, we will bear the expenses on the spot, and management is kind enough to grant the sanctions also, and I'm giving her free education, though she has to pay JVD, but I allowed her to give free education as long as she is in our campus. And moreover, our managements, they are coming forward and helping in all aspects. So the other day, our management secretary and treasurer, they have seen the performance and they were surprised, and immediately they have declared, so this much and like that. Gannapatra treasurer family, Gannapatla. So from that family, annually, so immediately he came and asked, so what is your this one and this? So she has given expenses. So immediately they have sanctioned 10,000 on the spot and 5,000 rupees from the abroad people, American people. They had get together. So he had asked me to convey this program, conduct the program. We arranged it, and people from America, they have come forward and given some amounts. Yesterday they wanted to pay, and possible. So they have already given something like donations. So it is so she is the ambassador for the KGRL College of Pharmacy. I wanted to convey the message. ఓకే, ఎవరెవరిలో ఎక్కడెక్కడ టాలెంట్స్ ఏముంటాయో తెలియదు. ఫస్ట్ ఇయర్లో ఫస్ట్ టైము ఫ్రెషర్స్ డేకి వేసినప్పుడు ఆ ప్రోగ్రామ్ చూసి నేను సర్ప్రైజ్. ఆ రోజు నుంచి జస్ట్ ఐఎమ్ గివింగ్ మై సపోర్ట్ ఇన్ ఆల్ ఆస్పెక్ట్స్. ఓకే, థ్యాంక్ యూ వెరీ మచ్.
థ్యాంక్ యూ భారతి. Now it's time for scientific sessions. Myself K. Devi, and now I would like to invite Dr. D. Nagarjuna Reddy Garu to give introduction about our resource person for the day, Dr. Sandeep Kumar Garu, Assistant Professor, Department of Regulatory Affairs, NIPER, Hyderabad.
So on behalf of Regulatory Department, sir, we welcome you, sir. and it's my pleasure to introduce, to give the important view of biodata on Sandeep Kumar Garu. Sir has acted in Department of Regulatory Affairs, National Institute of Pharmaceutical Education and Research, Balanagar, Hyderabad. And a few details about his academy is, Sir has completed his B.Pharmacy in Department of Pharmaceutical Sciences and also in Guru Jambeshwar University of Science and Technology, Haryana. And he has completed his M.Pharm in Pharmaceutical Technology, Naipur, Mohali, Punjab. And he has completed his PhD in Jalandhar, Punjab. Sir has acted as a lecturer in 2008-9 in Kurukshetra University and also as assistant professor in National Institute of Technology in 2014-17. And also his research associate in Department of Analytical Research, Theva API, Noida in 2017 and 21. And also he has acted as assistant professor in Department of Regulatory Affairs and is now pursuing in National Institute of Pharmaceutical Education Research, Hyderabad. In view, he has completed 30 publications and he has written 2 book chapters and 1 book, and he has attended so many international conferences. Sir, with this brief biodata, I welcome you, sir, to our college, and also we are eager to hear your speech, sir. Thank you.
A very good morning. I think it's not afternoon right now, is it? So I am not trained to be formal talk. I'll be little bit informal sort of. First of all, I would like to, trying to be formal initially. Let's see how formal I can go. First of all, I would like to congratulate the administration of KGRL and for providing me this opportunity. Actually, this is an opportunity for me as such. You people have given me a lot of respect. That's fine, and it's an honor for me. But ultimately, opportunity is mine, and we are pleasure to be here to present NIPER as such and the Department of Regulatory as such, right? So let's see. Please, can someone can start my PPT which I have shared. I would like to tell my audience that let's make this interaction, this session interactive, right? It should not be like that. Okay. Although I'll be catching you people also, asking little basics, right? Let's see how interactive we can make it. Mouse. Back हो सकता है. Although my topic will be related to impurities, how I have selected this topic will be based on what my experience was, a previous experience. Based on that, I have selected this topic. But before going to topic, it's our mandate that I need to introduce NIPER Hyderabad initially. Somebody can help me? Yeah. Initially, it's our statutory mandate to introduce NIPER Hyderabad as such. So, NIPER Hyderabad has its unique vision and mission to be an excellent, to excel in global pharmaceutical education provider. And we have almost PG and PhD in all 11 disciplines. And we have almost 100% placement because we have an location advantage, because we are in Hyderabad and it is a pharmaceutical hub. So every company is keen and eager to join hands with us so that they can take, because ultimately, our admission system is like that. Only those students which has qualified GPAT can only appear for the NIPER entrance exams. So we get cream of pharmaceutical sciences. So our students are best among many students all over India and competitive in international as well. So till now, almost 1,000 PG students we have passed and 53 PhD students has been awarded. Let's talk about little bit its inception. I think it has been incepted as KGRL Pharmacy College has incepted in 2007, and later on it progressed till 2021, in which 4 new departments has been introduced, and the newest among the newest department is regulatory affair. This is our total contribution in terms of publications, patents, MOUs, and recently we got NIRF second rank. So this is our achievement in 2022. We got NIRF rank, and we are proud to say that we are second in number, but we are just 0.04 points lesser than the number 1 point, that is Jamia Hamdard. So we are equivalent to number 1 in terms of NIRF institute in pharmaceutical field. So one more opportunity I would like to share here, that NIPER Hyderabad very specifically is providing an internship to B.Pharm and M.Pharm students. So those who are interested can see this web page. Can you see this page is not working? IT team? Yeah. So NIPER Hyderabad is providing an opportunity to be in part of NIPER for few months. Internship for 2 months and 6 months they are providing in various departments. Not in regulatory affairs till now we are not providing as of now, but in any other department we are keen to, and we are receiving, because it's a mandate from the Department of Pharmaceutical, Government of India, that we, being the premier institute in pharmaceutical sciences, should give the exposure of the other students also. For that, with very little amount of fee, we are welcoming the students from outside NIPER for maybe 2 months or 6 months. So coming to Department of Regulatory Affairs, we abbreviate this term as DORA, Department of Regulatory Affairs. Here, although I am the only regular faculty, rest faculties are from industry. They are supporting day and night, almost like regular faculty from various industries like Novartis, Biocon. These guys are working day and night, and till now we have 2 batches. In June 2021 it was initiated. Now we are running with 4th semester in 2021 batch, and the new batch has arrived in 2022. So as of now we are having 6 PhD students in regulatory affairs and almost 20 students in MS in pharmaceutical regulatory affairs. So about me, I think Dr. Nagarajan has given my introduction very briefly, but still I would like to emphasize here that how my journey bring me here. So I have done my Bachelor in Pharmaceutical Sciences from Guru Jambeshwar University, Hisar, Haryana, followed by MTech in Pharmaceutical Biotechnology from NIPER Mohali, and then I did my Then I immediately after completion of MTech, I joined Kurukshetra University for 6 months as an assistant professor over there, and then I continued my PhD in NIT Jalandhar for almost 6 years in Department of Biotechnology. And then I continued my work over there as an assistant professor on contract role for almost 3 years over there, and taught very basic subjects which I was just touching in pharmaceutical sciences, like molecular biology, microbiology, biochemistry, etc. And there I learned how the teaching is done, right? Because in every scenario, basics are repeated once and once, again and again. So here in this session, I'll be touching the basics also. I'll be trying to touch the basics of whatever basics are there. We will be asking the question from that point of view also. And after doing a PhD, I did 3-year experience, and from there I just moved to that is Teva. Teva API is an industry which is number one globally number one in generic manufacturing. There I was in Department of Analytical Research as an analytical chemist over there, and there I got touch with the ICH guideline related to qualities. So from there I got opportunity to be a part of NIPER Hyderabad, and there I demonstrated my aspects of ICH guideline which I learned during my stay in Teva API. So you can see my profile is a little bit hybrid, you know, moving from pharmaceutical, then pharmaceutical biotechnology, then core biotechnology, then moving to analytical chemistry, right? And then I moved to regulatory affairs. So point here is to mention that regulatory affair has no bar, right? So regulatory affair, if you are from very specific biotechnology, you can go in that, because there are some regulations involved in that, biologicals related to that. And if you are in analytical chemistry, yes, regulations are always there. And if you are going in chemistry, CMC is also having some limitations which is being regulated by the regulatory affairs provided by the global regulatory agencies. So regulatory affair has no bar. We are accepting the students from any field. I am having students, PhD students from pharmaceutics, chemistry, medicinal chemistry, analytical chemistry, even in analytical chemistry. So wide range of students we are accepting. So so what is regulatory affair? All many dignitaries has simplified the term regulatory affairs, although Mr. Patel will also be telling in detail, but still I would like to emphasize here that what is regulatory affair, that is it is a representation of quality, safety and efficacy, right? So regulatory affair want every people in this world to be safe with reference to drugs. So whatever activity is there, that is being regulated by the regulatory affair as a professional or as a regulator itself. So in each and every aspect, all these informations are required so that people who are taking the drugs in this world should be safe. So coming to these are those objectives which I have mentioned right now. So ultimately this is the seesaw which we have to capture, being as a regulatory affair professional or regulator. In every circumstance we have to check with respect to risk to benefit ratio, right? Benefit should always weighing the risk, right? Overweigh the risk. So in this scenario, regulatory affairs focus is being regulatory affair students, you might be knowing why the regulatory affair and regulation on the drug happened to happen. Something would have been happened that would have triggered such a situation. So first case related to drug regulation happened to happen with reference to sulfanilamide, right? Earlier sulfanilamide was very effective drug, although treating various infections, but as the manufacturer come to know its solubility in DEG, diethyl glycol, is very good, he immediately converted his formulation to DEG without any checking of the safety aspects, due to which what happened, almost many people died, 100 people died in USA, and after that safety aspect come into picture. And you also might be aware of another situation, that is Phocomelia thalidomide disaster, right? See, this drug was a miracle at its time, but no one was knowing enantiomers can also have such an impact that they can cause, one enantiomeric form can cause Phocomelia and another form is effective only. After that, two factors emerged in terms of pharmaceutical regulatory affairs. One is drug, that is enantiomerism, right? If some drug has different forms, enantiomers, it need to be checked and evaluated for safety also, and then followed by pharmacovigilance. How many of you know the term pharmacovigilance? Please raise the hands. Yes, pharmacovigilance, yes. You people must be from master's degree, right? So pharmacovigilance is nothing but reporting of ADR, adverse drug reaction. Have you ever reported any adverse drug reaction to any authority? Have you ever felt any adverse reaction in your lifetime with respect to drugs? I think everyone must have, but we do not know the way where it need to be reported, right? So that is Pharmacovigilance Department of IPC has an application. I think I have linked it with this. ADR, Adverse Drug Reaction Pharmacovigilance app is there. There you can report your whatever adverse drug is there, right? So all of a sudden I got loss of hair on my cheeks, right? And I immediately reported, and I reported with respect to paracetamol, because at that time I was taking paracetamol, but it is quite known that paracetamol is very safe drug. It cannot be, but my responsibility was to deliver the information to them. That could be due to paracetamol, that could be due to my COVID vaccination, or maybe 5G exposure we are getting nowadays. That could be due to anything, but point here is to report to the pharmacovigilance department. And it is not like it is a dead end situation. As soon as you will be reporting by this app, they will be calling you back within a day, 2 or 5 or 10 days, and they will be confirming everything, right? Whatever drawbacks are there, whatever information are left, they will be asking for that too. So it's about awareness, right? So the chairman of Indian Pharmacopoeia, he came to NIPER and he requested us to, he informed us and requested us to spread this information to all the world. So for that, I am presenting this thing in front of you people, so that the awareness should be there. Yeah, another case is Swan case, right? We all know thalidomide tragedy, and I think that previous elixir, sulfanilamide elixir, everyone knows, but it is even bigger case. If we talk about the drug toxicity has happened without any control over the drug, that is SMON, subacute myelooptical neuropathy disaster, right? Here the thing to emphasize is, okay, whenever one seesaw I have shown na, ke, benefit should be higher than risk. In this case, the people were treating themselves for this as an anti-flatulant, and it is not a big situation. One can bear that. But whatever the drug in the market for treating that flatulant, if the person is getting blind, it's a big case. So appropriate regulation should be there. So in Japan itself, 10,000 people got blind due to their treatment on the flatulence. So such a situation is happening throughout the world. That's why more and more regulation is happening on the drug, right? That's why its costing is there, right? So whenever some drug is costly, we should not blame the government, but it's about, you know, making people safe all the time. So regulatory affairs, as I've told you, okay, they are everywhere. They are into drug labeling, leaflets, they are into licensing, legal document, clinical trials, manufacturing, right? And regulatory affairs are the liaison between the regulator and the industries. So that liaison happens to happen with the regulatory affair professional. Every company has various departments, R&D departments. Among them, one department is regulatory affair department, and this regulatory affair department ultimately has an liaison with the regulatory agency, so that it can be interpreted appropriately, and ultimately every data is being received by the regulatory affairs department and communicated to the regulatory agencies, and whatever the questions regulatory agency has that comes back to regulatory affairs department of an industry, and they communicate to the respective department. I hope I am correct, Mr. Patel. Right, sir? So coming to very basic aspects, because ultimately we are here to be clear in every situation. We have to decide every situation, right? Whether something is drug or not a drug. We have to make everything a clear picture in white and black situation. No gray zone is generally there. So for example, if I take a definition from Drug and Cosmetic Act or New Drug and Clinical Trial, the definition of drug is written, anything which is used internally, externally for the cure, treatment, mitigation, diagnosis of the disease is called as generally drug. And whatever is not conveyed or completed in this definition, they have given some additional things, right? So that whatever things are there in the drug are given. So based on that definition, can you suggest me that this Head and Shoulder is a drug or not? It is a drug. It is mitigating any disease? No. It is claiming I am cleaning the dandruff. So it is not a drug, right? But if it claims I am treating the fungus which is causing the dandruff situation, so in that situation it becomes a drug, right? And now I will be coming to some other aspects also, which because you know it's a basic classes, so I start with, although I'll be going with the very specific session for which I was here, that is impurities in pharmaceutical drugs, that we will be coming. But can you tell me whether this Prega News is a drug or not? Why? Yes, come on, this is an appropriate time to interact. Right, this is an appropriate questions to be asked from the audience, and I think this is a stage where we can interact. Can anybody tell whether this is a drug or not, and if it is a drug, why it is a drug, and if it is not a drug, why it is not a drug? Prega News.
You will not find this type of things in future. So I is coming from NIPA, they have got very good opportunity to interact.
Chalo, let me tell you, some people will be thinking this is a drug because it is a diagnosing something, right? Because in definition it is written diagnosis. May not be treating, but it is diagnosing. But it is not diagnosing any disease, because pregnancy is not a disease, right? So it may come in medical device. So regulation with respect to medical device may implement, but regulation with reference to drug are not going to implement on that. So that's why we cover and we define which is where, right? So that level of understanding on words, on the regulatory documents, we have to understand and interpret and suggest our customer, whoever approaches us. Customer is not like we are taking money from anyone. Customer is like being in Department of Regulatory Affairs. Any other department can be my customer, right? And they may ask a question and I need to give them an appropriate answer based on my wisdom and knowledge, existing knowledge. So based on that, we define it. And you might be knowing term new drug, right? We all know what new drugs are. Those which has not been approved for safety, right? And in terms of with respect to Indian scenario, there is no term generic. I think you might be knowing the term generic, right? So in India, those drugs which are not new drugs are equivalent to generics of US FDA with respect to US FDA. But in any regulatory document in India, whether it is Drug and Cosmetic Act or whether it is New Drug and Clinical Trial, there is no term generic is being given. So we interpret automatically those drugs which are not new are generic, right? In this 4 definition, they are saying that these two, whether it is a modified or sustained release drug or some biological material, cannot be a generic one. They will always be a new drug, right? So such a definition is there very clearly. So we need to understand it. Okay, any biological things which are being produced in India are never going to be generic. They will be always new drug, right? So whatever rules implementing on new drug, whatever data required to get the marketing approval in India will be lying on that particular drug. So one another term is phytopharmaceuticals, right? Whenever phytopharmaceuticals terms comes, what is in your mind generally? Okay, some extract is a phytopharmaceuticals? No. Some plant itself is a phytopharmaceutical? No. Right, with respect to, because I am talking about a particular document, that is Drug and Cosmetic Act, and NDCT I am talking about. In this, they have clearly mentioned okay, the thing from the plant will be called as phytopharmaceutical if its fraction is being quantified or qualified, is being analyzed quantitatively or qualitatively. 4 analytes or 4 materials has been analyzed, and that particular fraction is called as phytopharmaceutical if it is not used in injection. If it is used as that is parenteral form, it will come in drug. So ultimately it will not be having a benefit of being phytopharmaceuticals. If it is an extract, it is not a phytopharmaceuticals, but it's a fraction whose 4 ingredients or 4 API or 4 chemicals are being quantitatively or qualitatively identified. Then only it is called as phytopharmaceuticals. So that such a clarity we should have while reading, because ultimately our work is not to have a skill on an experiment. Our skills lies in understanding the lines in the regulatory documents. So we need to understand those lines very clearly and understanding very deeply. So for that I just demonstrated here. This is the part of plant. Whether it is a phytopharmaceutical? No. Whether this exudate of a plant, whether it is a phytopharmaceuticals? No. But when we do extract, whether this extract is phytopharmaceutical? No. When we do its fractionation, in many of its fraction, one fraction is found and purified and analyzed with respect to its NMR, mass and everything, then it is called as phytopharmaceutical. So regulations lies on phytopharmaceutical happens to happen only when such a situation happens. And in the last, it is not for the injection. This is about the drug discovery process. You might be knowing in drug discovery process, chalo, leave this. This is not that important. I am coming to another drug development process. I think you all might be knowing. Any drug development process happens to happen and pass through pre-clinical trial and then followed by clinical trials, right? And clinical trials has 3 phases, right? And then it is followed by NDA, new drug application, and then followed by post-marketing approvals. So here I want to make one thing clear. Post-marketing surveillance and clinical trial phase 4, are they equal? Guys, please listen to me. It's a stage where we want to make something clear, right? Whoever is listening, either you listen or do not listen, right? If you are looking to listen very, very carefully. So can you suggest me post-marketing trial, post-marketing surveillance and clinical trial phase 4 are same or not? Yes or no? Yes? No. No. Phase 4 clinical trial are those studies, because you might be knowing what is clinical trial. Clinical trial are the studies in which we register the we register the patients and put them in direct supervision under the doctors, right? While pharmacovigilance or post-marketing surveillance do not register any patients. But still there are phase 4 clinical trials which are presenting, or initially say those companies which has completed their phase 3 clinical trial and presented the authorities all the data, and company says yes, your drug is safe, you can go for marketing, but you have to do all those studies which has not been completed, or it takes time. They allow us for the marketing, but still they seek for the data which is happening in clinical trial phase 4. In this clinical trial phase 4, they have to register a patient, right? They have to give a informed consent to the patient, and they have to take consent from the patient, and they need to have direct observation with the patient. So pharmacovigilance is different now, because I'm not registered, but I felt some adverse reaction, and I just reported to the authorities wherever I just suggested some app from IPC. So clinical phase 4 is quite different from the pharmacovigilance, right? Generally, the perception in anybody's mind is that clinical phase trial 4 is pharmacovigilance. No, it is different. So we can say that this is post-marketing surveillance. Post-marketing surveillance are of two types. One is clinical trial phase 4 and pharmacovigilance. Yes, you can segregate your situation in that way, right? So coming to main topic. So in this situation, I'll be asking some basic questions. So keep your pencil and pen or maybe some page with you. Perhaps I may ask for some calculation, right? So when I was in Teva for almost 4 years, right? So API is not an important part over there. Capturing impurity is an important part over there, right? Once in PhD, HPLC was installed, and that application scientist was showing me the baseline. He was zooming in, and ultimately found a peak. And he said, "Bravo, I can find an impurity." I just said, "Okay, we are concerned with API, then why you are finding?" But when I entered into Teva, then I just realized API can be, quantified in any scenario, but quantification of impurity is a issue, right? And there, the expertise of analytical chemist lies into, right? So whenever impurity comes, lot of questions from our upper authority come, right? Whether it is a residue, whether it is a organic impurity, inorganic impurity, whether it is a genotoxic impurity. So my mind was, with a lot of questions, right? And what happened in the next? Then I decided to take a deep breath and, do dhyan, and then your answer will come. So ultimately, I studied all the parts of impurities, what are different impurities, and how these things are controlled, and under which scenario, what decision one should take, right? So before going for upcoming slides, I would like to introduce some terminologies. Do you know what is the difference between drug substance and drug product? What is a drug substance? Do we have any facility if someone want to speak in mic? Someone want to speak about difference between drug product and drug substance? Yeah, sir, there.
Product is a finished product, and the drug is substance is one of the ingredients used in manufacturing of any drug.
Yeah, you are almost right, but in the second thing in which you are telling a drug substance is one of the ingredients, no. It is the main active API, right? Active substance is drug substance, right? And rest are excipients. Very right. Thank you very much. So, initially, earlier we were knowing KPI. These terminologies are being used interchangeably in such a scenario where such a discussion happened to happen. So initially we should know, okay, what is drug substance and drug product. Drug substances are the main active ingredients of the any finished formulation, and rest excipients. After mixing of excipients, it is called as drug product. And do you have any idea regarding reagents versus catalyst? Difference between reagent versus catalyst?
Reagent is one of the chemicals which we use in the reaction, and catalyst increases the rate of reaction.
And ultimately it comes out of the reaction and being reused, right? Lot of applause for such a nice answer. So we can see very clearly from this scenario, okay, if we do not use catalyst and reagent 1 and reagent 2 mixes with each other, and it takes an energy barrier, while if we take a catalyst, then it follows very low energy barrier and ultimately reaches to the product and releasing the catalyst out of the scenario, and it is being reused. So ultimately reagents are in excess and catalysts are in very minute quantity in any reaction. Do you know the difference or meaning of ppm? Parts per million and percentage we know, right? If I tell you, okay, if something is 0.05%, right, so how much ppm it would be? That's why I told you people to keep your pen and pencil, calculate and tell. 0.05% is how much? Percentage is the part in 100 and ppm is a part in 1 million, right? So 0.05 is in 100. If we multiplied it by 10 to power 4 and multiplied it to 10 to power 4, then it will become, I think, 50 or 500, right? 50 or 500. So it will become ultimately ppm in 500 perhaps. A zero ka difference hai, please adjust it. So coming to another part, I think we are getting short of time, so I should finish it a little quickly. What is the full form of MDD? Maximum daily dose. And what is the full form of TDI? Total daily intake. And what is the full form of PDE? PDE. Permitted daily exposure. Great. And TTC? Threshold of toxicological concern, TTC, right? AI is acceptable intake. So coming forward, I'll be going to upcoming slides. Impurity we know, right? It could be organic, solvent or inorganic. In this discussion, I'll not be touching with the inorganic part, but in case of organic, how it arises, it is arising from any degradation of any chemical reaction and process related impurities. So it could be API degradation, it could be a API excipient interaction, API residue interaction, API container interaction, or in process related excipient interaction, starting material, byproducts, intermediates, reagents, and solvent could be organic or inorganic, whatever it is. So classification of impurities, inorganic impurities, we can skip. Right, now I'm focusing here for the solvent impurity. As per ICH Q3C, there are 3 classes of impurities, right? One which is called as solvents to be avoided. Until unless you have a great justification, you should not use these solvents, because these are either toxic to health, and these are either toxic to health or hazardous to the environment. So based on that, these are the things. If they are being used, they have the limit, right? So they have the limit. For benzene, if you are using, it should not be more than 2 ppm, right? 2 parts in 1 million part of your total drug, right? And similarly, these have the limitations. But ultimately, for using this, you should have an appropriate justification. Okay, we have tried each and every solvent present in this world, and we are not able to work without it. So this is first classification, and next classification is solvent to be limited. Means these are the solvents which can be used, but with some limitations. And here the term comes as PDE, permitted daily exposure, right? So here you will be observing PD is 4.1 for acetonitrile, and its concentration is 100 time multiplication, right? So I'll be discussing how this 100 time is coming, and also I'll be asking, or I'll be telling you what PDE is exactly. So we have, I have just presented few examples of the solvents which has been used or selected by me, although it's a big list in ICH guideline. And for giving an example, so I selected only 4 over here. So class 3 are those solvents which is having very low toxic potential, but if it is low toxic potential, that doesn't mean we can use it to anything. It is also having some limitation that 50 mg per day equivalent to 5000 ppm or 0.5% with respect to option 1. We will be discussing what is option 1 and option 2 are. So coming to permitted daily exposure, right? This is a question to be asked generally. A regulator or any interviewer who is in industry, they are asking what is PDE, and in PDE another terminology comes out, that is NOEL, right? PDE is permitted daily exposure, means these are the quantity or these are the concentration of any solvent which can be used in any drug preparation, right? So in how this PDE is being calculated, it is having a formula, that is NOEL multiplied by weight adjustment divided by F1, F2, F3, F4, F5, right? So there are many factors involved into it, and I'll be giving an appropriate example how PDE can be calculated. First term is NOEL. Do you know the full form of this? No? No observed effect limit. Right? Means if you are treating any impurity or any drug with any animal, right, then it is that concentration, till that we have not observed any toxic effect, right? So that is that concentration, and its alternate is LOEL, that is lowest observed adverse effect limit. Means it is the lowest concentration while the study. Sometimes these are being replaced, right? So that's why I'm placing it here. LOEL, it is the concentration of any drug which is showing the adverse effect, lowest observed, right? So because using of NOEL and LOEL is having impact as a factor 5, right? So we will be discussing that too. So coming to different Fs, one is a factor on account of extrapolation between species. If we are doing this adverse effect studies in mice, this factor goes 12. And if you are doing studies on dog, then its factor, this factor goes to 2, right? So ultimately, more this factors value ultimately is going to reduce the PDE, right? Reduced PDE is more safe, right? Safer situation. So one factor is species, another factor is in human individual variations are there. So as a fixed value 10 has been added here as F2. And F3 a variable factor on account of toxicity studies, term of exposure. Means if your study on animal is short term, then this factor goes 10. And if you have studied for long term, maybe half life of any animal, then this factor goes to 1, right? Because ultimately, if you are giving the drug to animal for longer term, there are chances that adverse effect may be surfaced, right? So ultimately, the value increasing here is going to reduce PDE. F4 is the severity. If toxicity is being observed, what is the severity of that? If it is highly severe, then factor of 10 goes, and it is not severe, then factor 1 appears, right? And last is available factor may be applied if NOEL was not established. There are some situations, there are NOEL either you do by yourself, right? That's fine. If you do not have NOEL and you are following some existing information available in any database, in that case, there are chances for that any particular drug NOEL is not available. Instead of LOEL is available. In that case, you can calculate PDE with LOEL, but F5 would be 10. Right? F5 would be 10 if you are using LOEL instead of NOEL, right? So this is a representation of a drugs. Okay, what is LOEL and what is NOEL, right? Here, this point is showing this is therapeutic effect level, right? Because ultimately we do not want to present that concentration which is not showing any therapeutic level. So we will be determining NOEL or LOEL with respect to the therapeutic range only. So ultimately, this lowest point, it is showing no adverse effect, right? And if you have that data, that's fine. If you do not have that data, you can calculate the PDE with LOEL also. But if you include LOEL in that scenario, you have to put F5 factor as 10, right? To be more safer side, right? So if I say for calculation of PDE, if I say permitted daily exposure is NOEL into weight adjustment, yeah, the formula is same, but it is saying the study on mice was done for 13 weeks, NOEL was found to be this much, and no severe toxicity was observed. So what would be the PDE? If you put NOEL 48 here, weight for a normal human being is 50 kg, and since F1 would be 12 because it's a mice, not a dog. If it would have been dog, it would be 1, right? And F10, F2 is fixed 10 because variation among individuals happens to happen, right? And F3 is short-term studies because it is 13-week studies, and in case of mice, what is the age of mice generally? 100 days or 200 days? Whatever it is, but if it would have been 100 days or 50 days or more than that days, then this factor could have been 1 only. But here the factor is 10 because it is 13-week study. F4 is 1 because it is showing that no severe toxicity was observed. Toxicity was there, but it was not severe. Severity level could be if it is teratogenic, severe most. Carcinogenic, severe most, right? If it is itching, not severe, right? So based on that, that factor is 1, and since here NOEL was used, then F5 becomes 1 here. So ultimately, if you calculate, PDE come out to be 4 mg per day, right? So this is how it has been decided at each and every factor and every situation. Right, and another terminology is used, that is concentration, right? PDE is the permitted daily exposure of particular solvent or impurity which can be given to human directly. But what is the concentration? We can have that thing in drug itself, right? So that can be calculated by this formula, that is 1000, because the difference between PDE unit and concentration is different, right? There we use term mg per ml, mg per day, and here we are using term ppm, parts per million. So in that, so that this ppm can be calculated by multiplying by the factor of 10, because after multiplying with 10, this mg will become ultimately day by day cancel ho jayega, and ultimately it will become microgram per gram, because ppm is equivalent to 1 microgram per gram, right? Getting my point? So how that I was telling here, okay, generally, in general, 4.1 you multiply it with the 100, it will become a concentration, right? But there I am telling, okay, it is a factor of 1000 is being multiplied. From where 10 factor came, it is coming with respect to option 1, right? If your substance, drug substance is having a dose of less than 10 gram, so in that case, every drug substance, for every drug substance, it would be calculated by dividing it by 10. So ultimately, the factor of 100 multiplies. Getting my point? So any PDE, you just multiplied in general case. It's most of the cases. If you multiply PDE with directly 100, it gives you the concentration of the drug, which can be concentration of that particular solvent, which can be used in your drug preparation, not more than that, right? So it becomes a limit. And another option is option 2. If your MDD, that is maximum daily dose, is more than 10 gram per day, in that case, real time value will apply with respect to this particular equation. And if it is your drug product, then only option 2 happens to happen to calculate all those things, right? So this is a table which represents ultimately, because it's a formulation, so option 2 will implement here, right? Not option 1. So here it is a representation. It is showing K 3.68 mg per day is being used. So it is a passing. It is having no issue with respect to regulators to allow acetonitrile in this particular drug product, because ultimately drug substance was having 800 ppm, but you might be observing for ACN it's 410, but ultimately in formulation its weightage is very less, and ultimately it is coming up with this 3.68 mg, and in that PDE required is 4.1, which is less than that. So authority will allow us to go ahead with such a quantity of the solvent in your formulation. If I am going far away of time, please let me know. Okay. So another is organic impurities. As I have told you people, since it's not in any reaction, if you carry out any reaction, it's not like you will be 100% striking your appropriate location of your reagent, right? For example, if you are targeting, if your drug is at meta position, is making with meta position addition, but ultimately 100% will not be happen, and few percentage of ortho or para position will also happen in such a reaction. So these things are called as reaction byproducts, and ultimately it will become an impurity in your reaction, right? So how to do that, it's a chemist specialty or chemist skill, but ultimately what regulator or what regulatory affair professional will suggest in any situation we need to limit this impurity below some value, right? So such a direction from the ICH guideline I am going to discuss in next slide, right? And point here initially, okay, in the method development, okay, I am just discussing this one. Okay. So I'll be discussing this last point and I'll be closing, and will give an opportunity to hear from your side, so that I should make this session fruitful. So ultimately coming to this, in any situation when API is being given and analyst is trying to develop a method, right? But when he do not have other thing to separate in HPLC, you might be knowing what is the function of HPLC. It separates various compounds, right? And quantitatively as well as qualitatively also. So if that analyst do not have any impurity at hand from which this API should be separated, how can he develop such a method, right? For that, they have an option, or they have an option ke they degrade their drugs forcefully with respect to base, acid, heat, right? And ultimately whatever breakage will happen with forced degradation, there are chances in their reaction they can generate, right? So initially that person, the analytical chemist, can develop their method based on those available products with the forced degradation. So you can see here this particular drug after forced degradation are being appeared. So ultimately this process of developing method happens to happen and increasing because in any process new additional impurity comes into pictures. So in any process reaction 1 happens to happen, 2, 3, 4, 5 and many impurities comes into picture. But it is not mandatory that we should include those impurities in our final specification, because reason is some justification is there. Because if you see, if you have the data of purging efficiency, process trend data and control strategies, in that case we can remove those impurities from the drug specification. For example, in the start of any reaction, impurity concentration was 1, right? And in reaction 1 it is same, in reaction 2 it goes 0.5, in reaction 3 it reduced a lot, and in the final formulation it was not present, right? When it was not present in final formulation, no need to give this impurity as a specification to the product, final product. And similarly, if it is happening with this situation and it is giving 0, so no need. But there is another impurity which was present in the final formulation at 0.08 concentration. That is a trigger, right? Either you have to specify it in your final formulation that this impurity we are having in our formulation, right? Or else you can control this impurity in your starting material, right? You tell your supplier, okay, I'm asking for the starting material in which this impurity is less than 3, right? If it is less than 3, it will be less than 0.04 and it is acceptable without any specification, right? So this is called as control strategy. Another control strategy, I can give an example here. If your formulation is degrading with sunlight, right? In that case, what could be the control strategy? You are not throwing all your formulation away and all the research. Instead, you will be labeling, store the drug in a dark environment, right? Or amber colored container, right? So such a strategies can be put forth. So I'll be discussing this only because due to time constraint, and if you need, I can share these slides which are very long. I can share with you people, no issue in that. But I'll be ending with this slide and upcoming link available here. So in ICH, I think Q3A or Q3B is there. It is related to impurities, organic impurities in drug substance. There is written over there, okay, if your maximum daily dose is less than 2 gram per day, then your reporting threshold is 0.05%. Means you have to report any impurity whose concentration with respect to main API is 0.05, you have to report it, right? And if your MDD is more than 2 gram per day, then your reporting threshold reduces, right? And similarly, identification threshold, does someone know what is identification threshold? Identification threshold, it's a concentration, or you can say it's a limit. Threshold is something, limit, above which something happens, below which something do not happen, right? So it's a threshold above which one has to identify the impurity, characterize the impurity, know the structure of impurity, right? If you have the concentration below that, no need to go for its identification, right? So such a limit is there, such a threshold is there. If your limit, your impurity is less than 0.1%, that means you need not to identify it. Getting my point? And another is qualification threshold. If you are having impurity more than 0.1, right, and you have identified your structure, you can keep it, right? No issue in that. But if it is going above that, you have to qualify it for its safety, right? You need to check for maybe mutagenic activity, carcinogenic activities, right? And similarly, these things also changes with respect to more than 2 gram per day. But this is not as such very straightforward. Okay, less than 2 gram, 0.1. There is another term, that is 1 gram, 1 mg per day of the molecule. So for that I have prepared an Excel sheet which will be telling you the clear environment, right? Can you type 0.02? 0.2. 0.2. It's not happening. It's not moving, na? Oh, view only. Chalo, leave it. It's not working. I think some technical glitch happened to happen, but I would like to tell you, okay. In this scenario, it is not straightforward. Like, let me go once again there. So, chalo, I'll try to explain this. If you go by 0.2, do some calculation. If you go by 0.2, what would be the percentage of 0.1? If MDD is 0.2, what would be the percentage 0.1? 0.2 means 200 microgram. 200 microgram, 0.1% would be, I think, 0.2 microgram, right? So that would be less than this. So in this scenario, identification threshold will be 0.1, right? And similarly, if we calculate about this, that would also be less than 1 microgram. So percentage will be the same here. But if you go above 1 gram, right, calculate 0.1% for 1 gram, please calculate, it would be 1 microgram, right? For that, still it will be equivalent to this percentage, but if you go by this, it will be, I think, 1.5 microgram. And here, 1.5 microgram is more than 1 microgram. So here, this point applies, which is less than the percentage of 0.15. So I think with the interest of time, I am ending my session over here. And if you have any queries, whatever I have discussed, please come ahead. I'll be happy to answer. Few students have seen, have learned, I think, because they have made some eye-to-eye contact too, and I really appreciate their effort. And because, you know, in such a big scenario, direct interaction is not possible many a times, because mic may reach up to you, and there are some person is available to give you the mics or not. It happens to happen, that's fine. Those who have query can have an interaction of the dais also. I'll be happy to interact with you people. Thank you very much.
Thank you very much, sir, for your valuable knowledge and experiences sharing with us. And I now would like to invite Chairperson Dr. Sridhar Babu Garu to say few words about this.
Good afternoon to everyone. So it is a very excellent presentation by Sandeep Kumar sir. So the subject is somewhat rigid. It is just like pharmaceutical jurisprudence. I have seen so many faces, so it is difficult to understand, but Sandeep Kumar sir has delivered excellently with good examples. What is the necessity, need of this regulatory affairs, and also the impurities. So what is the role of impurities? So I think it is a valuable presentation and a knowledgeable presentation by this Sandeep Kumar sir towards the students. Thank you.
Thank you, sir. And I would like to invite our co-chairperson, Dr. Mohan Gandhi Garu, to say few words.
First of all, I would like to thank Sandeep sir for giving an excellent presentation in this regard. He has started his lecture right from the history aspect. The quote that he has made about sulfonamides. It happened some 200 years back, some early 1900s like that. But same thing happened some few months back in Delhi. So when you are in need of medicines, when so many pharmaceutical companies are rushing like mushrooms, there is always a problem with quality. So to cope up with the quality, we generally require stringent regulatory guidelines, and SAR has given very good examples how to deal with the impurities, how to deal with the guidelines. So I would like to congratulate sir, as well as thank you for a wonderful session, sir. Thank you.
Thank you, sir. And now it's time to felicitate our resource persons and chairperson and co-chairpersons. And I would like to invite our director sir and principal sirs and APTI members, vice president and president sir and secretary onto the dais. Thank you, sir. And now felicitation for our chairperson, Dr. Sridhar Babu Garu. sir. And felicitation for co-chairperson Dr. Mohan Gandhi garu. Thank you, sir. And now it's time for second scientific session. I would like to invite Dr. D. Nagarjuna Reddy garu to give introduction about our second resource person for the day. Sri Patru Patel, Regulatory Affairs Specialist, Dr. Reddy's Laboratory Limited, Hyderabad.
See, Mohan Gandhi was my student at the Vishnu College of Pharmacy. When I joined in 2007, he was my first batch student. I taught him this industrial pharmacy. Nataraj garu. So, Pavani. She is my direct student here, B Pharma and M Pharma in KGRL College of Pharmacy, alumni of KGRL College of Pharmacy, direct student, and he was my faculty member here. Later on, they got married, and Vasavilo recent chesaru. Now with the VB Institute of Pharmaceutical Science Godlavaleru. Okay, so very efficient person. So chala baga cheptadu lectures avi. Noora. Run. See, because all audience are there, very useful. ఈయన గురించి ఎందుకు చెప్తాను, నా స్టూడెంట్ ఇదంతా ఫ్యాకల్టీ మెంబరు, తర్వాత పిహెచ్డి కష్టపడి చేశాడు. ఓకే, జేఎన్టీయూ హైదరాబాద్, and he is a good resource person also. He gives a lot of lecture. Last time we had conducted an analysis on subject. These two guys were the speakers here. Okay. So first national conference in పెట్టానంటే APTI తో టైఅప్ తోటి ఫస్ట్ నేషనల్ కాన్ఫరెన్స్ అని పెట్టానమాట. దట్ వాజ్ ద టైటిల్ గివెన్. ఓకే, సో బికాస్ ఆఫ్ దిస్ రిసోర్స్ పర్సన్స్ అండ్ చైర్మన్, కో చైర్మన్ అంతా తెలిసిన వాళ్ళు కాబట్టి జస్ట్ ఐ వాంటెడ్ టు గివ్ ఇంట్రడక్షన్ టు మై ఆల్ డెలిగేట్స్ టుడే ఇయర్. థ్యాంక్ యూ వెరీ మచ్. ఓకే, థ్యాంక్ యూ భవాని. సో ఏమా నిన్ను మర్చిపోయినమ్మా, వీళ్ళిద్దరికి పుట్టిన బిడ్డ. ఓకే, రైట్. వీళ్ళిద్దరికి కూడా నేను ఓపెన్ ఆఫర్ ఇప్పుడు చెప్తున్నా, వీళ్ళిద్దరు కూడా ఓపెన్ ఆఫర్ ఇక్కడ, ఎనీ టైమ్ దే ఆర్ వెల్కమ్. సార్ ఒప్పుకుంటుంది, సార్ కూడా నేను చెప్పగలను, ఐ కెన్. ఓకే అమ్మా, సో లెట్ us ఎంజాయ్ ఇన్ ఏ ఫ్రెండ్లీ మేనర్. ఎందుకంటే అందరూ తెలిసిన వాళ్ళే. మొత్తం నేను ఇప్పుడు నరేంద్ర చెప్పారు నా గురించి. ఐఎమ్ ఇన్ టచ్ విత్ టోటల్ ఆల్ ఇండియా లెవెల్ ప్రిన్సిపల్స్ అందరితోటి కొంచెం యాక్టివిటీస్ అంతతో ఉండటంతోటి ఎవ్రీబడి లైక్స్. అదంతా మనకు కొనుక్కుంటే రాదు, బట్ ఇన్హెరెంట్ క్వాలిటీస్. ఓకే, రైట్, ఆల్ ది బెస్ట్. సో షల్ వి కంప్లీట్ ద సెకండ్ సెషన్?
So it is my immense pleasure to read a brief biodata of Patru Patel garu. He is working as Regulatory Affairs Specialist in India, RI team, Dr. Reddy's Laboratories Limited, Hyderabad. He has done his post-graduation specialized in Pharmacology Department in 2010. Overall, 13 years of experience, of which 12 years at vast regulatory experience with various pharmaceutical organization and regulatory affairs function in Pharmaleaf, Biocon, Kinapse and Wokhard. Handle on that regulatory requirement for the development, commercialization or post-approval phase of various products such as new drugs, biologics and biosimilars, non-prescription drugs, OTC and cosmetics. Additionally, on legal and policy matters as well as NPPA. So it is my immense pleasure to introduce about yourself, sir, and please start your session. So now I request Dr. K. Nataraj Garu to act as chairperson, sir. And I request Madam, Head of the Department, Pharmaceutics and Pharmaceutical Regulatory Affairs, SVCP, to act as co-chairperson.
You are writing slide. So before start of the session, I'll just tell you a story. When a drop of raindrop falls on different places, it gets associated and it adds value. When it drops or get it caught with our hand, we can use as washing or drinking. If it falls in the gutter, even we don't use. And the same drop, when it falls on the oysters, it will make a pearl. So the session, the information from your teachers or information from any place, it is coming as a same raindrop. Now the receiver will be you, how you will be receiving, whether you want to make this as a pearl or it is just wastage like drain water. So it all depends on us. Now to begin, so the time, considering time constraints, I will be briefing and I would request you all to ask any question in terms of regulatory, gain whatever is your doubts are there. So overview on Indian drug regulatory system. So today's session will be on these regulatory authorities in India and regulatory acts and various regulatory procedures. So I'll try to be a brief on that. So you might be aware which is the regulatory authority in India to give approval for drugs. Very good. So you are making my work easy. So who will be heading that CDSCO? Yeah, be open. Right, wrong will be decided later. First we need to have a courage to speak. Then those right, wrong will come. So who will be heading? Yes, DCGI. Who is DCGI currently? Yeah. You are into regulatory affairs. Might be regulatory affairs team also here. There is a two terms. One is regulatory and other one is affairs.
So that affairs is current affairs on regulatory. So you must be knowing regulatory related everything, and it is a dynamic, like share market, how it changes. Similar way, regulations, rules, guidelines will change. So I cannot be using the 1947 made act or rules, because if you read that book, you will not find a term biosimilar, a term medical device, gene therapy. Why? Because the regulatory profession is one that will go along with innovation. So whenever there is innovation happening, there will be a regulatory changes to handle that. So Ministry of Health and Family Welfare, under that CDSCO is there, and also Indian Pharmacopoeia Commission. IPC's role is two. One is at establishing Indian Pharmacopoeia monographs as well as pharmacovigilance. And there are other, like Ministry of Science and Technology, in that RCGM, so which deals with the biosimilar, biologics, vaccine products till preclinical. Preclinical means animal toxicology studies. And also DSIR and CSIR, this is required for R&D activities certifications. And at this Ministry of Chemicals and Fertilizers, Department of Pharmaceutical, under that NPPA, National Pharmaceutical Pricing Authority. So any drug in India which are available, medicine, if you are going and purchasing, all those medicines falls under 4 baskets. So one is medicine which is scheduled price. Scheduled price in the sense which is listed in National List of Essential Medicines. So for that, this agency will fix the price, and once they fix, for example, paracetamol 1 rupees, everyone whoever is marketing, they have to sell that drug only at 1 rupees or lesser than that. The second category is products which, medicines which are not listed under NLEM or scheduled. So there the agency will not fix the price, they will monitor. Monitor in the sense, if marketing company is increasing price annually up to 10% is fine. More than that, then there will be a show cause notice and they have to face music. Now the third basket is new drugs. This new drug is differ from the Drugs and Cosmetic Act. So this new drug, what it says, scheduled formulation. Like paracetamol is listed under schedule. If I am increasing strength 500 to 650, or if I am combining with other drug, listed or not listed, then that will be new drug as per DPCO 2013, and I have to get the price fixed by this agency. So this is the third. And the last one is para 19. This is something extraordinary power given by the central government to fix the price of the medicines which are not accessible to Indian patients because of its unaffordability. You might have heard cancer drugs, they have brought in this. There are so many medical devices they have brought in. So the information is available everywhere. You have to concentrate and learn on this, and today the information is flooded like anything. So you just have to visit these websites, you will gain lot of things. Now the next is Ministry of Agriculture, and in this what plant quarantine and animal quarantine. This also required for us if we are dealing with any product which is coming from the animal source or something to import, this permission is required. So the other one is Ministry of Finance. Central Bureau of Narcotics, you might have heard this term, CBN. So this deals with the narcotic drugs and psychotropic substances. The more stringent regulations are for controlled drugs, are nothing but NDPS. So there are two different ministries also there. I have not captured here. Ministry of Home Affairs. There the CBN will become NCB. So you might have heard Shah Rukh's son and other cases. Those were all under the NCB jurisdiction. So even if we are dealing with this product, we have to have the 0.1 gram reconciliation data also. And if without approval, if it lands on Indian territory, in the sense port, like seaport or airport, without permission or under permission, then there will be a direct show cause notice and non-bailable warrant to the authorized signatory persons for violating these rules. So the majority of our work related to the CDSCO and state licensing authority. There is other Ministry of Environment. This deals with Genetic Engineering Appraisal Committee for vaccine and biotechnology products. So, yeah. So you can see geographical representation, how it is. CDSCO headquartered at Delhi, and it has 6 zonal offices, north, all 4 direction, and after that they have added Hyderabad and Ahmedabad. The reason being adding those 2 are having the pharmaceutical companies there, and their role is, I'll explain the subsequent slide. Apart from that, sub-zonal offices. So these are like located in Bangalore, Jammu, Goa. So DCGI organogram. So there will be several divisions under CDSCO. If my molecule is biologics, it will go to biologic division. If it is a new drug, name NCE or subsequent new drug, it will go to new drug division. And if it is medical device, medical device. If the molecule is import and marketing, import registration division is there. So this is just brief structure of state licensing authority. It varies from state to state. So every state will have one drugs controller or drugs commissioner. Under that, deputy drugs or joint drugs controller, likewise terminology ADC and drug inspectors. And at the parallelly, there will be a laboratories directors for those testing laboratories. So you can see the functions, main functions of each of the CDSCO headquarters. Main function is approval of new drugs, approval of clinical trials, approval of medical devices. These all approvals, deals, or even any new drug, it will go to central CDSCO. Besides that, there will be like standardization or issuance of guidelines, notifications, circulars based on the regulators, like technical advisory boards or ministry. So these, and also delegation of certain powers to these zonal offices, sub-zonal offices. And zonal, sub-zonal offices, issuance of delegated permissions from the central licensing authority, like dual use NOC is used for import of raw material or not for medicinal use. Those are test licenses, inspection and recommendations on after inspection, like written confirmation, WHO GMP, COPP. whereas the state licensing authority deals with the issuance of manufacturing licenses, manufacturing test licenses, certificates like free sale certificates, COPPs, and other certificates to participate in tenders. Then apart from that, there are other activities like inspection of facilities, drawing up samples. They will come and draw the sample from any premises or pharmacies and they will send for the testing. And if there is reported as not of standard quality, then the letter will be issued to the company where it is manufactured. So those are the functions of the state licensing authority. So you are aware of all regulatory acts India perspective, DNC Act and rules, and NDCT rules, and Act will be drugs and cosmetic, and these rules will be amended from time to time. Why it is amended, like as I said, because of innovation or to meet any challenges, these rules needs to be amended to control on those aspects. So Drugs and Cosmetic Act 1940, it is effective from 1st of 1947. What happened? Before that, our Indian market completely dependent on the import of drugs. So what happened? Increased demand. So whenever there is a increase in demand, there will be a chances of compromise on quality. So they flooded our market with the adulterated substandard drugs. Then the R.N. Chopra committee has been formed, and based on that, this act promulgated. So act has 5 chapters and 2 schedules. So for details, you can even download from the CDSU website. So these are the definitions which I already covered. So if there is any questions, I will take on this. So how it will act? There are 3 main categories. One is at advisory roles. So Drugs Technical Advisory Board, Drugs Consultative Committee, and Subject Expert Committee, New Drug Advisory Committee, earlier used to be called, now it is Subject Expert Committee. And at the same time, there are other committees which I have not captured, Technical Committee. So these are advisory to the authorities for making their decisions very effective and in line with the regulations. Whereas analytical central drug testing laboratories, regional drug testing laboratories, there are so many government laboratories for different kind of molecule will be tested at different laboratories. Like for vaccines, it will be CDL Kasoli, whereas for biologics, it will be National Institute of Biologics, Noida. And executives, our licensing authorities and drug inspectors. So the entire act provisions of these act will be governing towards import, manufacturing, sale, and labeling and packaging. So this is drugs and cosmetic rules, and there are so 19 parts and various schedules. So Schedule H, Schedule G, you might have heard, those are the schedules. So NDCT rules, earlier it used to be called as Schedule Y. Now it is New Drugs and Clinical Trial Rules 2019, effective from the 19th March 2019. They have improved a lot in this, and they have added certain new things which is very positive steps towards the industry and also benefits to the patients, like orphan drug definitions and exclusivity and definitions of new rules and more clarity on the forms. So as any new drug will fall under any new drug. Anyway, we had a definition. So old drug is something which is, if it is approved in India and it crossed the 4 years, it will be old drug, except two cases. One is it should not be modified release dosage form or novel drug delivery system, and it should not be a vaccine, sera, biologic or rDNA product. So rest all will be old drugs. In case of old drugs, we don't have to get the permission from the DCGI. Directly we can apply to the state licensing authority and get the manufacturing license. Based on that, we can launch our product. So here the regulatory approval stages at various in development and commercialization of drugs. So development phase, there are two kind of test licensing required. First is manufacturing test license. So this test license will be issued by the CDSCO as well as state licensing authority in a different forms, structured forms. So after that, we will be taking the batches, 3 developmental batches, and also doing the toxicity studies, animal toxicity studies as per the NDCT rules. So once we complete, then we will be submitting our application to CDSCO for respective phase 1, 2, 3 in a sequential manner for NC. So these are all online portal. We have to just submit online as part of e-governance in India. All these applications will be submitted through online. And once we complete entire process, then there will be a marketing authorization phase. Here we will be submitting an application in a CTD module, which is India specific CTD module, through online with fees and all. After submission, there will be a proposal call for the applicant to present our proposal before the subject expert committee. These subject expert committees divided into 12 different therapeutic categories. If my molecule is like diabetic, it will go to endocrinology. If it is heart related, it will go to cardiovascular. If it is cancer related, it will go to oncologists. In these individual expert committees, eminent professors, physicians, pharmacologists from the renowned colleges like AIMS, NIMS, likewise KIMS, these. So what we will be doing, so we have to present our proposal before them. They will go through particularly on clinical aspects and non-clinical aspects. CMC will be taken care by the Indian regulators. So after going through that, if they are having any questions, if they are not satisfied with the data which we have presented, they will ask us to modify or do. If it is satisfied, they will give the recommendations in the like the proposal can be considered based on the clinical study or BE studies. So once we finish, then there will be a marketing authorization approval from the Indian regulators, that is DCGI. These are all online system. So SEC also, earlier it was face to face, because of COVID it is virtual. Now the same process is being continued as of now. So once we receive the CDSCO approval, our work is not over. Then there is a manufacturing license which is mandatory to launch our product. So manufacturing license will be granted from the state licensing authority where my manufacturing facility is located. If it is in Andhra, from Andhra regulators, Telangana, Karnataka, likewise. So before granting, they will come and do the inspection of the facility to see whether it is in compliance to the GMP requirement or not. That is called as Schedule M in India. And also the dossier which we have submitted, source document verification, whatever the details we are mentioning. So based on that, there will be a manufacturing license. After this, we can manufacture the batch and launch the product as commercially into the India market. So this is marketing phase, and the final phase is post-approval change. So post-approval change is compliance to the conditions. If you go and purchase any kind of shopping, if they give receipt, there will be a set of conditions mentioned on it. Although we do ignore to read those, the similar way here on permission or approval, there will be a set of conditions which needs to be complied, and the same conditions we are complying, so that will fall under post-approval change. Or if we are doing any amendment, scale up, all those activities will be part of post-approval phase. And submission of periodic safety update reports, which is required for that. So there are three phases: clinical, imported, indigenously developed, as I explained. So this, I believe you will be knowing various clinical trial phases, how many patients required. Normally phase 1 will be first in human studies. It will be approximately 20, the numbers may vary. Phase 2 will be for evaluating dose ranging studies. I might be having, but I have to have the ideal optimized dose. So they will design in a cohort of 5 patients or 6 patients, and then they will evaluate 2 mg, 8 mg, like increasing order. So whichever is effective, they will use. In the phase 3 clinical trial, this will be a confirmatory efficacy trial. If the primary objectives and secondary objectives of this trial meets, then only we can make the product marketing authorization application based on this clinical study report. So this is very critical phase. So these are requirement of toxicology at various stage. You can find in the NDCT rules also. So single dose, repeated dose, and duration, and subsequent toxicity studies based on the nature of molecule, dosage form required. So there are provisions in the NDCT rules with respect to exemption. If the molecule is already approved in US, Europe, ICH countries several years, then they may consider for same molecule, if I am developing in India, they may grant us the exemption for toxicity studies. So there are certain challenges, although provision is there. The expectation from the regulators, from the facility where I have taken this. So this I have briefed you entire process. So import and registration. If time gets over, please let me know. So import, if you want to import any molecule which is manufactured overseas, I will have to follow 3 steps: import, registration, and then commercial import license. So import permission will be in a similar way, like application submission, and then there will be a SEC, and then there will be a grant of approval in specific forms. After that approval, I will be filing for registration application in Form 40 for registration of oversized manufacturing site and product. So these, after submission, then there will be a grant of registration certificate from the CDSCO. Based on that, application in Form 10 for commercial import license. So once they grant the import license, based on this approval I can import, and the imported product will be get tested first time at CDTL Mumbai. Whenever it reaches to the customs, it will come any of the ports, either airport or seaport. Then I can launch the product into the India market. So manufacturing and marketing permissions. So these are the different forms which are applicable. So WHO's GMP certificate, these certificates will be issued by the state licensing authority. So if you have any question, please feel free to ask.
Any questions from audience? We conclude the session. Say yes or no.
Thank you, sir, for enlightening us with your experiences and knowledge. And now I invite our chairperson, Dr. K. Nataraj Garu, to give few words.
Thank you, Mr. Patel, and it's given a very wonderful because there is two kinds of knowledge usually is to gain the students. One is hands-on experience. Second one is what it is there in the bookish knowledge. Okay, so what Patel sir is told that Mr. Patel sir, because these things you cannot get in the books. Okay, so these are all to be what really happening that in the industry. He told very well that is okay, especially even as far as my concern that I learned today that is 4 years old drugs are not essential to take the licensing from CDSO. Okay, we have to take the license from the state governments. Okay, so some knowledge we can able to hear while attending for lectures, and while few can understand by reading books. Okay, so information how we gain, that is one of the important aspect. Today there is a widespread, we have very scope for all of us, but you can never replace Guru with Google. So they are all gurus, that is, they're sharing the knowledge, they're standing on the platform and they're sharing the knowledge. So there's from my bottom of the heart, I should very thankful, sir. So it is beneficial session, even though it is not in books. So what you told, that is really impressive, sir. Thank you, sir. Thank you.
Thank you, sir. And I invite our co-chairperson, ma'am, Dr. Lakshmi Prasanthi Garu, to few words.
Good afternoon, dear students. Sir, given the data in a simple and explain the complex data in a simple way, all of you studied this information jurisprudence. At that time, you feel very discomfort to remember the data, but he delivered that information in a simple and easy manner. Thank you, sir.
Thank you, ma'am. Now it's time for felicitation. I invite our director sir and principal sir and APTI members to felicitate our guests. Yes, sir. Thank you, sir. And felicitation for our chairperson, Dr. K. Natraj Garu. Thank you, sir. And felicitation for our co-chairperson, Dr. N. Lakshmi Prashanti Garu. Lakshmi. Thank you, madam.
So then the next program is the lunch follows in the lawn. Okay. For the faculty members, there is a separate counter on the left side. All the students can have their lunch in the right side lawn. Okay. So after the lunch, there is a session of poster presentation, e-posters and oral posters in the ground floor itself. Those who want to register, they can approach the registration desk in the entrance. Okay. Have your lunch and come back for the So 2 o'clock, the poster sessions, e-poster sessions, oral poster sessions will be starting in the ground floor. So all the students have your lunch and come back fast for your poster sessions. Thank you.